what it represents is this blue bar or blue curve that once established immunological memory remain more or less constant. Okay, for most infectious agents or for most antigens, right? This is what basically it highlights. Now, on the other, basically this axis here, so is talking about the response of antibodies. Okay, for example, if you dilute these antibodies in 1:2, for example, 2, 1:4, 1:2, whatever, right? And then reverse of this dilution, okay, you call as titer. Okay, so that means you can dilute serum to the extent of, for example, let's say 32 times, 64 times, 128 times, 4 times, and still it retains activity either to neutralize the, for example, virus, or retain ability to bind to that antigen. That is what you are looking at. So these are two things here. So ideally, it should have been represented in two different diagrams. Okay, but just to combine that, or just to actually show a comparative analysis on antibody response and memory response. So how do they basically pan out following vaccination? Okay, as you can see, the antibody titers initially after vaccination increase, reaches a peak, right? We are not even measuring the number of antibody forming B cells. We are just measuring the soluble product that you have in circulation. Okay, so you take out serum, and in serum you measure antibody against that vaccinogen, okay, whatever the antigen you injected. And over a period of time, then the titers of these antibodies decay. Right?
Fine, sir. And now I got it. Yeah, thank you.
So on this axis, basically what we are looking at, once you achieve this immunological memory, that it's not like it's going down again. Okay, so it remained more or less constant. That is what was emphasized in this plot.
So there is one question on the chat.
Could you please explain positive and negative selection again? Well, no, I don't want to, because you keep on coming to those things which I just have referred, and I need to discuss all those things as a real topic. Okay. So talk, basically, if you want any clarification on whatever we have discussed, not referred. Okay, I'm pretty sure we must have referred to almost every thing in immunology, but I have not really covered those things yet. Okay, so just wait for those lectures which are going to talk about positive and negative selection. If something is not clear from whatever we covered, which is actually written in slides, so you can ask those. Otherwise, we keep on blabbering about something, and I have not really talked about that yet, and you not understand, and then you'll say, yeah, I don't know, so what, whether it makes sense or it doesn't make sense. So why do I do that? Just to explain something which I have not really touched upon for the first time, right? I said that thymus, because I introduce these terms in context of these organs, these lymphoid organs, primary and secondary lymphoid organ, that bone marrow is the site where development of B cell take place, and development involves these two processes, positive and negative selection. That's what I just have said as of now. And same is the case with T cells, which occur in thymus. Okay. So anything which actually is written on slides and you want me to talk about, so I can do that. But not those things which I have perfunctorily referred to, right? Because there was a context when I mentioned or even named those terminologies. Anyways, I think now we can actually start.
Sir,
Yeah,
is Malto pairs patches part of tertiary lymphoid organs?
yeah. You tend to call them as tertiary lymphoid organs. They are not actually secondary lymphoid organs.
Okay, sir.
All right. So we were talking about these different immune cells and their known functions, right? And the last cell we covered was mast cells and their major role in releasing histamine, which is responsible for causing these allergic reactions or hyperinflammatory reactions, right? How exactly this is occurring? So again, we are going to talk about it. But just to, again, very briefly mention that this is because of the receptors expressed by these mast cells that we call as Fc receptors. And if the antibody ligates to those receptors, then basically this cross linkage of this receptors, Fc receptors on mast cells release these granules, and these granules are responsible for causing allergic response. Okay, so we were here actually there. Okay, the other cell type is NK cells. As the name suggests, these are natural killer cells. So what that means is they are naturally poised to kill either infected cells or transformed cells. Okay. Now, what are these transformed cells? These cells which are developing into cancerous cells. And why only these particularly two cell types which are responsible for this activity of NK cells? Okay. So, and that is more or less explained in this diagram here. Before that, so how do they carry out this function of killing is by releasing these lytic granules. Okay, so these lytic granules. Okay, and what is carried in these lytic granules is these effector molecules, which are also found actually in your cytotoxic T cells, like granzyme B, perforin. Okay, so these molecules are responsible for either making pores in the target membrane, and once these pores are created, so then they basically enter, and then they basically these granzymes, so they basically carry out the apoptotic processes in these cells, the target cells. Okay, now let's talk about this thing which is on the other panels. So let's say you have this target cell. Okay, so this, consider this as the target cell. Now, this target cell is having this MSC molecule on their surface, and because MSC molecules are present on every cell type, almost all of them, except for their lower expression on neuronal cells, right? So once these cells are infected, so these microbial derived peptides, so they are loaded onto MSC class 1, right? And those MSC class 1 loaded with this peptide are recognized by the normal cytotoxic T cells. Okay, you have this effector cell, target cell. This is cytotoxic T cells. You have this peptide. When you have this TCR peptide MHC, and the cells are activated, so they release their effector molecules and they try to kill this cell. Okay, this is a normal situation, right? When these cells are expressing normal level of MHC class 1. However, there are many infectious agents which downregulate the expression of MHC class 1 when cells are infected. And same is the case with cancerous cells. Cancerous cells downregulate MHC class 1 on their surface. Now, probably in that case, cytotoxic T cells will not be able to kill those cells so efficiently, right? So then as a alternative mechanism, so you have these NK cells which come into picture, and they are normally basically NK cells do not work if you have MSC1 class MSC1 expressing cells in vicinity. And why? Because MSC1 ligates to some of the molecules expressed by these NK cells. Okay, and this interaction gives negative signal to your NK cells. Okay, NK cells normally are not functional in the sense because MSC and the ligand expressed or the receptor expressed by NK cells, this actually switch off or turn down the function of NK cells. But as soon as you remove MSC class 1, so this negative break, this break actually is removed. And once this break is removed, so now there is another set of activating receptors which are more dominant. And as a result of this dominant interaction effected by the activating receptor, cells become activated and they start releasing their cargo, which actually involves these lytic granules and many more molecules. And those things actually are then entering into these target cells and they kill them off. Now, this hypothesis actually is called missing self, because MHC class 1 or even MHC class 2 are your self MHC molecule. Particularly for NK cells activity, if you have downregulation of MHC class 1, your NK cells get activated because then the negative regulation is removed and they are now actively engaged, able to kill these cells, right? So the mechanism by which NK cells work is called missing self hypothesis, and it was actually shown in infection conditions as well as in some tumors. Maybe I'll show you some slides, maybe in one slide to show actually this is how it was demonstrated. All right, but is that clear? How do NK cells function? So the mechanism of working by your cytotoxic T cells and your NK cells is different. Okay, the mechanisms of these two cells are different. Cytotoxic T cells, via expressing their TCR, which ligate to peptide, which is loaded onto MSC class 1, get involved in this interaction with the target cells, and in so doing, so they release its stuff, and the target cell is killed off. But as I said, there are conditions where MSC class 1 is downregulated on surface of these infected or transformed cells. In that case, cytotoxic T cells will not be able to function appropriately. And as an alternative a mechanism, your NK cells get activated and they try to remove these target cells. Because they recognize target cells once they do not express MHC class 1. Okay, this is called missing self activation mechanism by NK cells. All right, so now a little bit more about NK cells. So these cells, you could realize, work at the crossroad of innate and adaptive immune responses. One is because their killing mechanisms are very similar to what you have for your cytotoxic T cells, which are part of your adaptive immune response, right? Second is, so basically, then how do you classify them as innate immune, their activity, which is similar to those of innate immune cells, because they are recruited and they act very quickly in the response. And it's because these cells already are loaded with these preformed granules carrying lytic um, uh, effector molecules like granzymes, porphyrins, glycans, and many more, right? So all those things are already present in these cells. And whenever they get a chance to get activated, so they do it so quickly and try to get recruited in the response very early on. For adaptive immune cells, it will take some time. Okay, that is why NK cells actually are working at the crossroad of innate and adaptive immune responses. Their major function is in anti-tumor defense and also against some pathogens. Now, in an unmanipulated mouse, okay, so basically how these cells were discovered. Okay, so this is what is highlighted in this third point here. And this also tells you something about the biology of NK cells. In control animals, which were not infected, which were not injected with anything, okay, so they removed spleen cells, and surprisingly, they were able to observe anti-tumor activity in naive spleen. Naive means the spleen which were isolated from these organs in absence of any injection of an antigen or an infection, or in absence of a developing tumor in these animals. Okay, normal spleen. Normal spleen, if you actually basically disrupt and make single cell suspension from splenic tissues, and you probe using certain assays, so there was anti-tumor activity associated with these spleen cells. Okay. Now, what cells had this anti-tumor activity? So they were not T cells, they were not B cells, right? Because they depleted T cells from these samples using antibody or through different approaches. They also depleted B cells using a different way. So there are different ways of removing these cells from the sample that you have. Okay, if you want to know, so we can talk about it at some time point, but that's not relevant here. So just remember that you can remove B cells or T cells or any other cell type that you are interested in looking at the function of. Okay. Now, if T cells were removed, the activities remained. If B cells were removed, activity still remained. So now, around that time, so these two cells were the major cell types which are associated with tumor activity. Okay, in a normal spleen, rather in lymphoid organs, right? But the activity was still there. Further evidence came from these experiments which were performed in wild type versus RAG1 deficient or RAG2 deficient animals. Now, what is RAG? RAG is an enzyme. The other name actually is a recombination of activation gene. Okay. So this enzyme is required for generating a fully mature form of TCR or BCR. Okay, so you have two chains for your BCR, and you have rather four chains of your BCR and two chains of your TCR, alpha and beta. And for BCR, so you have heavy and light, and two of those. Okay, two heavy and two light. So this constitutes a BCR, and one alpha and one beta constitutes a TCR. Now, in order to form this TCR or BCR, so you require the activity of this enzyme. Now, if you remove by genetic depletion this enzyme from animals, this anti-tumor activity still remained. Okay, so what it tells you that this is independent of RAG recombinases. Okay, development of NK cell was independent of RAG recombinases. Okay, and they were not T cells, they were not B cells, and they were actually present in normal spleen of unmanipulated mice. So these are some of the now evidence that you have gathered to show what could be the origin of these NK cells, right? And that is why, because they were not classified as B cells, they were not classified to express any marker associated with T cells. So they were called as null cells to begin with. Okay. Now, in this slide, basically what it shows is the activity of NK cells was found to be crucial in tumorigenesis. Okay. So there are different models which you can make use of if you want to study tumor immunology. So there's some question. Okay. Can VTZ recombinase be also clear? Well, yeah. Well, VTZ recombinase. The other name of actually VTZ recombinase is RAG. Okay. This is exactly the same gene I'm talking about. But you don't call it as VTZ recombinase. You just say recombinase or recombination activation gene. So that's the full form. Anyways.
Excuse me,
Okay.
sir.
Yeah.
Sir, could you please explain why development of NK cells was independent of RAG once again?
Was independent of?
So the RAG negative mice, you said that it was independent and hence it was called null cells. It didn't involve T cells.
Yeah, well, this is what I said to begin with. Vishnu, so what do you want to try out?
Yes, sir.
So explain, she's asking something. So what's your name?
Sir, Aradhana.
Aradhana. Aradhana is asking something.
Yes, sir. So you told that RAG are basically recombination of activation gene, which is an enzyme required for fully formed, TCR and BCR. So if you take the mice and like produce the RAG negative, double negative, and measure the anti-tumor activity, it is still showing the anti-tumor activity. So that clearly signifies that, and from the earlier experiment we saw that like this anti-tumor activity is due to the NK cells. So this clearly signifies that NK cells development is independent of this RAG enzyme. So yeah, is it fine, sir?
Yeah, well, I think so. Aradhana.
Sir, just by this conclusion, can we say that it will be independent? Wouldn't
Well,
there be more set of experiments that should be involved here?
Well, so you are too early. So let me complete this slide first. Okay, but
sir.
as of now, whatever we have covered So and this is what he summarized, right? So what essentially he said was that even in absence of RAG enzymes, so these cells were present in spleens, and they were still able to perform anti-tumor activity. This is what he said. And RAGs are required for development of your T cells and B cells. And essentially, this is required for actually recombination of these different snippets, different segments you have in TCR and BCR. So we'll talk about those things in detail. Okay. Another evidence which came that NK cells actually are critical for providing defense against tumors was performed in these animal models where you can actually implant these cancer cells, right? And over a period of time, so these animals will keep on developing more and more growth of tumor. And you can measure the volume of tumor. You can actually measure the size and in what area basically this tumor is expanding, right? Usually you try to do it in a skin cancer model. And the simple reason is because then over a period of time, you can actually measure in the same mouse, so whether the cancer is developing or it has now regressed. Okay, you can use mesothelioma or any other lung cancer or any ovary cancer or breast cancer. So you can use any model. Okay, but for that, so the scheme has to be different. So you have to sacrifice these animals at different time points, and you have to actually calculate what is the volume at different time points and so on and so forth. But if you want to follow the progression of tumor in the same mouse, so you need to generate these tumors at a particular site, right? So that you can visually observe it, right? Now, what was done? So tumor was injected in these animals. Now, what these animals were, I'm not going to even talk about those because this is a little too complicated for you to understand at this time point. There are different strains of animals. They are inbreds. They are a knockout of different components. But just remember that if you inject these animals with tumor cells, and if these tumor cells actually are quite aggressive in their division, so cancer will keep on progressing in these animals, right? So tumor cells were injected, and if you follow in control animals, control in this case, it will be control immunoglobulin. Okay, so basically there are two groups here. One is, in one you inject an antibody which is control, isotype control. Again, I know I'm talking about something I have not yet covered, isotype control. But for the time being, just remember that this is the control antibody, and in this case, an antibody against an NK cell marker was used, okay, to deplete these NK cells. How exactly this depletion happened? Again, I'm not even talking about that. Okay, consider that you are able to deplete NK cells using antibodies, and as a control, you have to use another antibody which doesn't deplete these cells. Okay, and that should be enough to understand these. Okay, now if you monitor the progression of this tumor over a period of time in control animals where NK cell activity was there, so you have this development of this tumor, and it reaches to a certain level and then it goes down. And this is because this is actually quite some time after this injection, and then you might generate some kind of immune response, and then this tumor is regressed. Okay. Now, if you deplete NK cells, okay, if you deplete these NK cells, these animals, they kept on developing tumor unhindered. So this suggests that somehow NK cells probably are playing a crucial role in controlling progression of tumor. Because the only thing that was different in these two groups is depletion of NK cells. Okay, and so actually this data is collected from different papers, so that is why you have the same numbers and so on and so forth. Okay, just to show how the experiments were done to demonstrate whether or not NK cells are playing a role in anti-cancer defense. Okay, in another case, so the same thing. So the tumor was injected, and in control animals, so it develops and then it regresses, and you are comparing the activity of your CD8 T cells and NK cells in controlling tumor. Okay, and you can clearly see that both NK cells and CD8 T cells are important in providing anti-tumor activity. Okay, because in both these cases, even if either you deplete NK cells or you deplete CD8 T cells, the cancer keep on dividing, but not in wild type situation where NK cells were there, where CD8 T cells were there. Okay, now what molecule is required for which is produced by either NK cells or CD8 T cells? What role does it have to play? And one of the molecule which is produced by NK cell actually is interferons. Okay, rather interferon gamma. Now, exactly how interferon gamma works, there are different mechanisms. I'm not talking about those. Just to show that if you neutralize interferon using an antibody, so again you get the same phenotype. So from these experiments, it's kind of suggests that like CD8 T cells, NK cells are also contributing to defense against tumors, and this was dependent on interferon response. All right. Now, so this is again a different model. Now you can see that here, so in control, so it's actually at the very basal line, right? And in these animals, the control actually is also showing some level of cancer development, right? So now, this basically is explained by what animals were used. Okay, so in this case, they must have used probably a different strain or different for example, nude mice, maybe reg deficient animals, maybe skid animals, right? But in this case, it could be wild type. That maybe if you use a less number of these cells in wild type animals, so you don't even develop tumor, because this tumor is controlled by the immune response, which are actually sufficiently induced in these wild type animals, so you don't develop tumors, right? But as soon as you deplete either NK cells or CD8 T cells, so you start getting more and more tumor growth. Okay, so this slide highlights the role of these NK cells along with CD8 T cells in providing defense against tumors. Okay. Okay, so now if you talk about the different markers, although this is too much information for you at this time point, but just to keep you abreast with. Okay, so these are the markers which are actually found to be present on surface of human NK cells. Okay, And these are the markers which actually were shown to be present on surface of um, mouse NK cells. Okay, so you have many of these chemokine receptors. You have some specific molecules which are present in mouse, which are not present in humans. And in humans, so actually you have this molecule CD94 NKZ2C. So this interaction, okay, so actually which is very critical for controlling the activity of these NK cells. Does it recognize? It can recognize many of the elements derived from these viruses, okay, like herpes viruses, chikungunya virus, HIV, hepatitis B virus. Some of the elements of these viruses can be recognized by this receptor. So in addition, so you also have many of these cytokine receptors present on surface of these NK cells. IL-12 receptor, IL-15 receptor, IL-18 receptors, right? And this is these are the molecules which you find to be present on memory cells. Okay, and there is a reason for that, because these cytokines, if are sensed because of the presence of these receptors, so these cells can sustain. If you don't understand too much from this slide, so it's all right, so don't worry. Okay, this is just to highlight that there are a lot of shared feature between human and mouse NK cells, and some are unique features, right? They express chemokine receptors. They also express a lot of cytokine receptors on the surface. Okay, particularly in mouse, this molecule CD49H, okay, was shown to recognize M157 protein of this mouse cytomegalovirus. Now, why I'm actually highlighting this? Because using this molecule, 157 of human cytomegalovirus and MCMV infection, so a very nice paper was written where they showed that NK cells indeed have features which are shared between between innate and adaptive immune cells. Rather, this paper actually was the first one to show that NK cells have all the characteristics that you will find for your adaptive immune cells. And what are those? Specific recognition through expression of these molecules on the surface. For adaptive immune cells, we call them as TCR or BCR. But in NK cells, so this molecule was the responsible for this whole thing, CD49H, right? Second thing, once you have specific recognition, these cells expand in number dramatically in the acute phase, okay? And third, there is a contraction phase, okay? So if you let's say assume you made 100 of those cells from one cells in first few days, so that out of 100 cells almost 95% are going to die. This was also demonstrated for NK cells, right? And then whatever the remaining cells that you have, 5 to 10% of the cells, okay, so they can be recalled if you have the same antigen given to these animals again. Okay, so these are all the features of adaptive immune cells, which were also shown to be true for NK cells. All right, so these are many more molecules. If your life was not easier in previous slide, so okay, so I'm pretty sure this is not going to be even easier if you look at the number of these molecules, but you don't have to remember or even read these things. Okay, so there are many more molecules which are expressed by these NK cells. Okay, adhesion molecules, activating receptors, inhibitory receptors. So I think this is important because we talked about the role of activating receptors and inhibitory receptors in missing self hypothesis, right? So just go through these, go through this slide. Uh, Otherwise, this is only for academic purpose, uh, much beyond your level of understanding in this course. Okay, so in advanced course, yeah, you have to remember all those things. Now, one thing actually that you have to remember is, how do you identify these NK cells in different strains of mice? One of them actually is an antibody against this molecule NK1.1. Okay, if you find NK1.1 positive cells in these black 6 animals, so you can call them as NK cells. Okay, so the other antibody that you find is this DX5. So this is a clone of the antibody. This is the clone name. Okay, if you use this antibody in biopsy mice, and if you stain some positive cells, you can actually say that these are NK cells. Okay, that is why I have actually highlighted these two things. So these two markers are, these two antibodies actually are responsible for detecting the identity of your NK cells in black 6 and biopsy animals, and they express these activating and inhibitory receptors. All other things we are not going to talk about. If you want, so you can just go through it, that they don't express any of the molecules associated with the T cells, right? They have NK1.1 positive, 122. This is actually a cytokine receptor. CD3 negative, DX5 positive, again in biopsy, so you can easily see 122 positive, right? So these are basically combination of marker you have to make use of to identify NK cells. Functions, function associated with NK cells are what we call as antibody dependent cell mediated cytotoxicity. So that is one of the function, and why antibodies are critical for the function of NK cells, because they express a lot of these Fc receptors. And when we talk about the structure of antibody molecules, so then you will realize more and more why ADCC contributed by NK cells is critically involving the role of antibody molecules. Okay, but for the time being, remember ADCC is associated with NK cells. Molecules which are formed by these NK cells are highlighted here: perforin, granzymes, Fas ligand, trail, interferon, gamma, TNF alpha, and in some cases even anti-inflammatory IL-10. These cells exist in different subsets. Some of these subsets can be predominantly involved in cytolysis, and some are in producing cytokines. Mode of function is through missing self hypothesis, and licensing of this NK cells is provided by self-MNC molecule. Okay. This is the paper I was talking about. Just read through it so you understand what I was saying. Just read it for 1 minute. Read it? So essentially, this is the abstract of this paper where they showed that NK cells have adaptive immune features. Okay, basically what they are saying is if you infect these animals with this virus, uh, by recognizing this L49H receptor on their surface, NK cells expanded almost 100-fold in spleen and 1000-fold in liver. They underwent a process of contraction and generated a pool of memory cells, and these memory cells could then further be recalled efficiently with the same ligand. That means M157 of this LCMV, of this MCMV virus. Okay. Now, this paper actually was published in Nature in 2009, and the title is "Critical Players of what is this? Critical Players of Innate Immune System." Rather, this is the title: "Adaptive Immune Features of Natural Killer Cells." If you want to read more about this paper, read it. So it's very interesting paper, which for the first time showed that NK cells have all the features which are also associated with your adaptive immune cells. Okay, so very quickly we'll talk about this slide. So what essentially it tells you is that using flow cytometry, you can identify these different cell types that we initially said can be classified based on this staining. Basically, if you make a smear on the slide and you stain with this dye, you can mark them as neutrophil, basophils, eosinophil, lymphocyte, monocyte, and so on and so forth. Okay. This slide actually highlights that these are the markers you can make use of for identifying whether you have T cells, B cells, macrophages, granulocytes, megakaryocytes, or erythrocytes. Okay. This is again for your knowledge. Why do NK cells remain in non-lymphoid organs? What are these? What is this? Yashvi, so ask this question. So I do not understand what you're saying. Well, they are found in non-lymphoid as well as in lymphoid. They are actually found in spleen, and they are also found in liver, lungs, and elsewhere. So yeah. So what did you want to ask?
Like, do they follow the secondary expansion in non-lymphoid organs as well?
Well, actually, that has not been very clearly elucidated. Well, at least not in this paper that I just referred to. Okay. But obviously, if they are present in liver, so chances are that and if the virus is also present there, so they might expand locally because the cytokines are also present, right? And you have the ligand to activate them. So now, if you say that expansion of these cells in non-lymphoid organs, so this actually is one of the canonical feature of a memory cell of both T cells and probably in NK cells as well. We call them as tissue resident memory cells. Okay, that's not, but clearly, so it was not shown in this paper that whether this expansion actually is happening in non-lymphoid organ. Maybe they were expanding in spleen and then they migrated from spleen to non-lymphoid organs. Okay, so it's already 10:55. I think we need to stop here. So a little bit more of this slide will cover in next slide, in next class, and then we'll continue from there on.
Excuse me, sir.
Yeah.
Sir, could you please go back to the slide where NK cell diagram was shown of mice and human?
Okay. So you don't want to attend your next class?
Sir, just a quick doubt.
Yeah.
Sir, here in the bottom left here, I can see that there is a clear-cut demarcation of red and yellow, whereas in top
Which
right
one?
it is quite diffused.
Which one?
Sir, here the red and yellow, the slide that has been divided in two halves of red section and yellow section. So in bottom right, it is quite clear-cut demarcated.
I'm colorblind. Well, I don't know what you're saying. So why don't you actually name some of those things which I can actually look at?
Sir, actually 3/4 of the NK cell mouse is covered in red portion, and
So
that is,
this one,
yes, sir.
okay.
And sir, towards the right hand side, the top right is quite diffused. The red and yellow colors are quite diffused.
Yeah.
I think it is trying to suggest that there is not a clear-cut demarcation between memory and memory-like
Well,
receptors.
yeah. Well, obviously. Well, yeah. So this slide actually was much before then. Actually, even this paper came out, which showed that this might have a memory phenotype and so on and so forth. Well, see, somebody has to get a clue. Why would these cells be considered as memory cells and so on and so forth? Because they express these cytokine receptors, so one can always sense that they must be proliferating or maintained because of the cytokine which are actually being produced by other cells, right? And that might be responsible for their maintenance in the host, even in the memory stage or in memory-like stage, right? Where you don't actually require the TCR stimulation for maintenance of memory cells, or BCR stimulation for maintenance of memory cells. It's just the cytokine-dependent proliferation. It's a very slow turnover without involving specific receptor.
So, sir, does this happen only in humans? Because in mouse, it hasn't been shown.
Of what?
Sir, that
For
that
for maintenance of these cells?
yes, sir.
Well, actually, it has been shown in mouse using this virus. This is actually a mouse virus, murine cytomegalovirus. So this paper that I referred to was actually all done in mice. But more or less, you have similar features in human NK cells as well.
Okay, sir.
Yeah. Okay, so this is not to say that now you can start calling NK cells as adaptive immune cells. No, because one of the very critical feature of for adaptive immune cells is they have a broader repertoire, and here they were working only with one of these receptors, right, which sense the presence of this viral antigen, right? So they don't have 1 billion antigens which can be recognized by these NK cells. Their repertoire actually is also very limited in order to provide this immunological memory and so on and so forth. Okay, but nonetheless, so they actually talked about the features which might be ascribed or attributed to their being working at the interface of adaptive and innate immune cells.
Right, sir.
Yeah. Okay, so I'm